In the name of Allah, the most beneficent and the most merciful. Assalamu alaikum, dear students. I hope you all are well and fine and enjoying your studies. I welcome you in Pakistan International School Types virtual learning class, and this is class of biology for grade first year. Dear students, we have started chapter number 2, that is biological molecules, and in that we have seen up to now lots of things actually about carbohydrates, its classification, about proteins, its classification, and we have started in the previous topic lipids. Today, and we have seen its first class, that is acylglycerol. Today we will see the next class of lipids. So you can see here in this flow chart that lipids can be divided into 3 groups: simple lipids, compound lipids, and derived lipids. So in simple lipids, we have seen acylglycerol, in that they are the esters of fatty acids, in which the other classes is fats. We have discussed fatty acids here, and then we are having waxes. They are simple lipids. Other type is compound lipids, in that esters of fatty acids and alcohol contain other groups also. There are fatty acids which are having some other groups in their esters, which are attached with them. So they can be, we can say phospholipids are there, glycolipids are there, as well as we are having lipoproteins. So here we will discuss phospholipids only. Then we are having derived lipids. In derived lipids, we can have terpenes, steroids, prostaglandins, and cholesterol, in that they are composed of hydrocarbon rings and a long hydrocarbon side chain. How their structure are made up of, we will discuss after some time. Okay, if you see this one, that what are lipids? So just take a review of the previous lecture, that lipids are macromolecules. They are made up of fatty acids, monomers, especially fatty acids are there, and their function is to having structural and supportive role for the cell, as well as energy storage and cell signaling as well. Like if we talk about cholesterol, cell signaling, there are the lipids are non-polar, we know, and they cannot interact with water. It means they are hydrophobic in nature, like fats and oils, waxes as well. So here we can see 3 types which we have discussed like before: simple, complex, and derived. So triglycerides, which we have talked about, they are simple one. And if you see this one, waxes, they are also simple one. And if you see steroids, then steroids are having other example of steroids like cholesterol and phospholipids. They are complex, and steroids are the derived form of that. We will discuss in a few minutes. Okay, dear students, so let's start waxes. The type which is given in your book. Okay, so waxes are actually highly hydrophobic compounds. And if we talk about these waxes, so there are 2 types of waxes. There are natural waxes. They are also simple lipids, as well as there are synthetic waxes as well. Natural waxes, which we can get from naturally from different type of natural things. Definitely they are, we can say they are found uh, from the bees wax, which is found in honeycomb, honey, or bees just not make what we can say honey, but they also make wax with that one. Lanolin, that is found or obtained from the sheep wool, that is taken, like actually lanolin is secreted by the sebaceous or skin glands which are present under the wool in the skin of a sheep. As well as cutin, we know that cutica, it is the wax which is present on the cuticle of the leaf surfaces of the plants, which protect them from evaporation, transpiration, as well as that make the plants leaves and like all the plants stable to stand upright in the harsh or strong sunlight and outside environment. Then we are having suberin. Suberin is found in a cell wall of endodermis of plant roots. Actually, so that is derived from the Latin word suber, which means cork. It means this present in the cork and cell wall, which makes them stronger and reduces the evaporation. And as well as this is having the protective functional. If we talk about the plant's roots, which can protect the plant's roots from the bacterial invasion also. So all these are chemically inert and resistant to the atmospheric oxidation, like they just keep the plants or keep like the organisms safe from the oxidation process, as well as from bacterial invasion also. Waxes are actually esters of long chain of fatty acids and long chain alcohols. Okay, so now we will see synthetic waxes that are generally derived from petroleum and polyethylene. Actually, because they are synthetic, they are made from these products. These are actually raw materials, and we just derived them or made them in the industries from petroleum. Like we are having what, paraffin wax, WFIN. Okay, paraffin wax that is made from petroleum, or that is synthetic waxes can also be prepared from or synthesized from polyethylene, um like from the polymerization of ethylene, and that wax made by direct polymerization of this ethylene, but under special conditions and which are provided to them. And they consist of uh, because these are synthetic, so they consist of lots of some other groups inside it, and like a mixture of different groups, and they are long chain of hydrocarbons can be there, like alkanes, and alcohols can be there, mixture of aldehyde and ketones can also be there, as well as fatty acids. So they are in different ratio. They are collectively or as a mixture, they are present in them. Okay, aldehyde and ketones we have discussed before in carbohydrates as well, with carbonyl group C double bond O. If one side H is present, so it means Another side, long chain of carbohydrate is present. Carbon is present. And here we can say in ketones, both of the sides, in place of H, what is present? R, like long chain of both of the sides, a long chain of carbon can be present. Here can be R, and here can be R also. So these are the some groups which are present in the synthetic waxes. With the mixture, they are like synthesized, and like we can have the examples of paraffin wax, which is used to make definitely candles. Wax paper, we know that wax paper is utilized as the lining in some for the lining in the molds and the baking dishes, as well as lots of other purposes are there. Lubricants and sealing material, they are very, very important regarding the machinery, machines, cars, and different type of machines which are running, and to reduce their friction, so we use this type of things. Now we will talk about the next group of lipids, that is phospholipids. So what is going to be happen here, that it is a type of complex or compound lipids, definitely, and commonly occurring are derived from phosphatidic acid. Like with phosphatidic acid, which we are going to combine to form phospholipids. Phospho means phosphorus, and that is lipids. With the fatty acids, like as we have talking about triacylglycerol, what we have seen, that you can see this is the head of glycerol, in which three types of fatty acids are, three chains of fatty acids are present. So this is called triglycerides. But in phospholipids, with this head of glycerol, just two chains of fatty acids are present. Two chains of fatty acids are present, and in place of the third one, in the head, what is present here? Phosphate group. If you see in the next picture, then you can understand well. So phospholipid is formed when phosphatidic acid combines with one of the four organic compounds. Which compound? This is choline, that is a nitrogenous base, and a ethanol amine, that is amino alcohol. That name ethanol, that is alcohol, and amine means amino alcohol. N-isotyl toll suggests that there is alcohol, and that is amino alcohol, and serine, that is amino acid. If a phosphatidic acid molecule is more similar to diglyceride, as I have told you before. This is, if we talk about just this one, okay, so this is diglyceride. When with this glycerol in place of this third fatty acid chain, phosphate is like making bond with that one, so that is called phosphatidic acid. And with that phosphatidic acid, if choline is present, so that is called phosphatidyl choline, or that is called lecithin, so that is phospholipid. And this phospholipid here can be like, if it is choline, so it is lecithin, and if it is any other like ethanol amine can also attach here, so there can be the different compound. So you can see this structure, um, you can see this structure, that is phospholipid, and this phospholipid is The main constituent of membranes, plasma membranes, if we just remember the fluid mosaic model. So in that, what happened, you can see this one, then this one, and this is the long chains of this, and here this, and like this one. There are two parts of phospholipids, as we have seen here. The one is this one is known as head, and this is tail, because this is long fatty acids chains. So here the head, and here also the head, and in between of these, in the centers, they are having tails pointing towards one another. So these are this is the structure of fluid mosaic model of cell membrane, and here for lipid bilayer is present. So that lipid bilayer is formed by this phospholipid. Okay, so the upper one, that is polar hydrophilic head. So this is hydrophilic, that's why it's pointing out towards the outer environment. This is from inner to the inner environment. This is inside of the cell, this is outer of the cell. So it means that they are hydrophilic, like they this is, sorry, inner one. So they are present with the water, they make bond with the water, but inside this fatty acids chain, this is hydrophobic tail, and they are repelling the water. So you can see the structure with this one, that they are there is double bond. That's why this chain is slightly bent to the other side. Here is glycerol, this yellow one phosphate group, and this one is choline. So that is called lecithin. And these are the major constituents of plasma membrane. Next, we will talk about terpenes. So terpenes are the types of derived lipids. Like this is derived lipids is another group of lipids, so like another class in which terpenes are included. So they are synthesized form of a 5-carbon building block known as isoprene unit. If you just see this one, 1, 2, 3, 4, and this one 5. So these are 5-carbon atoms which are just making the bond in this form, zigzag form. So like this. So here is methyl group, and here is hydro group, and here is double bond, and here also double bond. You can see like this. So this is the simple structure. So you can see this unit condenses like it to make polymer or a macromolecule with the combination of lots of by lots of different ways, but with the continuation of this isoprene unit. This is isoprene unit. When two isoprene units will combine like this with one another, so like it will make the bonds like this again. Here will be methyl group will come here, and hydro group will come here. So this will, when two isoprene units form, this units form a compound that is called monoterpene. And this monoterpene is present in menthol. You know that menthol, like it is present in peppermint, and mint, which give sharp aroma as well. 4 form diaprene, like if one unit of this one and like this one, for example this one, we just say it monoterpene. So when the two more will join, so this will call diaprene. Yes, diterpene. This one is monoterpene, and these two are diterpene. Like this, if six will form, they will call triterpene, and then after this one, we will say polyterpene. So diterpene example is vitamin A and phytol, which is we'll talk about in chapter number 4 as well, chlorophyll tail. So phytol is the chlorophyll tail, which is made by this diterpene compound, and triterpene is amberine, and that is actually act as analgesic in a different like plants. Natural rubber is polyterpene, like lots of units of isoprene are over there. So let's talk about steroids. Steroids are lipids of high molecular weight, and they can be crystalline as well. So if we talk about one steroid unit, so that consists of almost 17 carbon. You can see here 17 carbons are there, but they are arranged in 4 hydrocarbon rings, and these rings are attached in this form. If you can see here on the diagram, 3 rings are having 6 carbon atoms, and the 4th ring is having 5 carbon atoms, and the other are outside of that. The 2 are outside of that. So the length and structure of the side chains that extend from these rings distinguish from one steroid to another. But if we talk about cholesterol, cholesterol is the steroid, and actually this is the precursor of a large number of equally important steroids. Large number of steroids. In those, we are having bile acids which are present in the like gallbladder, male sex hormones, this is testosterone, female sex hormones, they are called progesterone and estrogen. Actually they are made up of cholesterol, and bile salts as well, which come from the gallbladder, which emulsify fats, and as well as vitamin D is also made from a cholesterol, which helps to regulate calcium metabolism. So they all are steroids. Actually, what happened here, we can see that here methyl group is attached, like CH3 is attached here, and CH3 is attached here. There is also methyl group is attached. And the very important that here at carbon number 3 OH group is attached. Due to that, that is called cholesterol. So this name all is said to be, yes, OH group is attached with that one. If we just talk about the cholesterol structure, so actually this is at carbon number 17, the long chain of carbon is attached. Yes, you can see here like this. Long chain of a carbohydrate or carbon is attached. And this is the specification of cholesterol, that here the cholesterol, if the long chain of carbon is attached with carbon number 17. So cholesterol is very important, that is having the structural component of the cell membrane because it gives buoyancy, fluidity, and it gives the strength to cell membrane as well. Okay, let's talk about the last group, which is mentioned in your book, that is prostaglandins. And for prostaglandins, actually that is present almost in every mammalian tissue and act as local hormones. They are derived from arachidonic acid. Arachidonic acid is actually tetra unsaturated 20 carbon fatty acids, so in which 20 carbons are attached, and in that tetra unsaturated means here 4 double bonds are present in these 20 like carbon fatty acids. If you see in the previous we can see here the previous page, previous topic. So you can see here arachidonic acid, here 1 double bond is present here, number 2, 3 and 4 is present here. So that is tetra unsaturated carbon 20 fatty acids. So okay, if we talk about its function, that is very, very important function is done by prostaglandin hormones, which are actually derived from our phospholipids or lipid bilayer of plasma membrane. You can see here, here and here. So here we can say the lipid bilayer of the cell membrane, of the plasma membrane. So from here, by enzyme, this prostaglandins in the form of arachidonic acid are going to be synthesized. Okay, so when this arachidonic acid is synthesized, then this is changed into different types of prostaglandins, and they are having very wide role in our body. So some reduce blood pressure and some raise the blood pressures. So this is having very vital role in immune system as well. If we just take a look at the immune system regarding prostaglandins. So number one, they help to induce fever. It increases prostaglandins. Some prostaglandins increase the temperature in the body, so body temperature gets higher. So this is the alarming situation and good sign for the body to tell, to inform that something is going wrong in our body. So let's treat that. So number two, it causes inflammation in that required area where the injury can be, and as well as it intensifies the sensation of pain, increases the pain in that area. And next, number four, some prostaglandins, what they do, they increase or like collect or aggregate the platelets at the required area for making the blood clots over there, for to stop the blood, to clot the blood. And like prostaglandins, which are like synthesized by hypothalamus, because temperature regulating center is present in hypothalamus, so they cause a fever. So all these things is very, very important regarding regulating our body processes. So here, like four things are there, like redness, heat or raising the temperature, pain increasing, and swelling is over there at the required area. So it increases the blood blood dilation, or we can say the blood vessels dilation, by which it permits the increase of blood flow to the affected area to give more oxygen supply, as well as these blood vessels are having more permeability to escape white blood cells and leukocytes to that area. And what can happen, as well as we can say more and more white blood cells when move to that place, so the place the white blood cells start to to decrease the amount of pathogens over there, or to kill the pathogens, so the area or the place will be inflamed and red over there. So against that, because like for the time being, this is a good sign, but for longer period of time, this is dangerous for the health, because lots of healthy cells can die in the specific area if we are not gonna treat that one. So we use analgesics, or that is very common, that is aspirin. That is actually anti-inflammatory drug. It reduces the inflammation, as well as analgesic, like it reduces the pain, as well as antipyretic, it reduces the fever as well. So how does it act against prostaglandin? That it inhibits that enzyme which synthesizes prostaglandin by releasing or by synthesizing arachidonate or arachidonic acid. So aspirin, it inhibits this enzyme, that is prostaglandin synthetase salicylate, uh, which inhibit the metabolism of arachidonate or arachidonic acid by another pathway that is called lipooxygenase. And what it does, lipooxygenase enzyme, it changes 12 hydroperoxy to 12 hydroxy. Oxy, like what is its full name, like what is the acid which is changed into arachidonic acid, that is 12 hydroxy 5 8 10 14 eicosapentaenoic acid. So this acid, like the same compound, but what happened here is peroxy and here is hydroxy. So its like formula is C20H32 32 and O4. This is 3 actually. So this is 12 hydroperoxy 5 8 10 14 eicosapentaenoic acid. But when one oxygen is removed from that, so then it will become C20H32O3, that is 12 hydroxy. So by changing hydroperoxy to hydroxy, arachidonic acid is going to reduce, its synthesis is going to be reduced, and then prostaglandins are decreasing, and then inflammation will be and treated, as well as pain and fever will be treated, and it is reduced. So dear students, with this topic of prostaglandins, we have completed our, we can say, the classification of lipids. We have just completed that one. So inshallah, we will continue uh, with the new topic of biological molecules in the next lecture. Wish you all the best. Allah Hafiz.